Atomic Era Diagnostic was founded in 2019, and our aim is actually to improve health by proteomics.
Atomic Era, we exclusively work with Bruker instruments, so we use the timsTOF Pro as well as HD both for our plasma and tissue workflow.
Since we're using the timsTOF HD, we are actually quite happy to see in almost all projects that we can find more proteins and we can quantify them very precisely. So recently we did also a project on B cells, and we were quite happy to see that more than 10,000 proteins could be identified in 60 milligradients.
My name is Andreas von Daimling. I'm a MD, I'm a neuropathologist, and my primary work is getting the right diagnosis of brain tumors. I have a special interest to develop tools which allow me to get things right, to do better than I can do myself on the microscope. I'm convinced that mass spectrometry will become a standard in future tumor diagnostics. From the same analysis with mass spectrometry, we can find neoantigens, we can find new peptides, fusion peptides. From the same data, we can read out that specific proteins are highly overexpressed, which I'm sure will in the future be very helpful in evaluating the aggressiveness of specific tumors. Assessment of multiple things simultaneously in one analysis, which we currently need several different complicated analyses for, will be the major advantage of mass spectrometry.
Tissue proteomics, the samples we work with, they can go in diverse directions. So it can be that we have a project with pathologists, for example, where it's about looking more at the spatial resolution, so you can also cut out different regions on FFPE samples, look at the stroma versus cancer regions that are more invasive or less invasive, and this really goes into maybe identifying a new subset of cancer, or it can be about identifying understanding the different mechanistic behind the development of cancer. And on the other hand, it can just be about normal proteomics discovery, where you get a large study of samples of healthy versus control, and then you try to identify what kind of potential biomarkers could help you to better define a particular cancer type.
Plasma proteins, they can be between different individuals 10, 100 or even 1,000 times different. This means we have a huge variation, and how you can actually tackle this variation is by measuring large-scale cohorts. So you need a high throughput to measure a lot of samples to deal with this high biological variation. The Bruker timsTOF instruments, they give us actually the robustness that we need to have a high throughput and to be able to measure a lot of samples in a very reproducible manner. The combination with the EvoSep system is quite nicely, because this combination gives us really the throughput of like 100 sample per instrument combination per day.
We can load more sample on the instrument, and that will give us a higher proteome depth when we analyze complex samples with a high dynamic range. And also the digitizer helps us to have a more precise quantification. To be simple, it just means better results. So often we also think about how many details do you want to communicate to the customer, how many details do they know about. But I think in simple words, it's just getting more precise and more proteins that you find.